All right, welcome. Today we're going to be talking about Chapter 9, and this lecture will be divided into 4 parts. And Chapter 9 is basically all about some of the biochemical pathways in the cell that allow it to harvest energy from sugars. And these reactions are generally associated with the mitochondria. You may remember the old cliche, right? The mitochondria is the powerhouse of the cell. That's where ATP is being produced. And so what we want to talk about in this chapter is, well, what are, and just at least in an overview, look at an overview of what are some of those biochemical pathways that allow cells to produce ATP. How does that work? And as I said before, that's usually referred to as cellular respiration. We need oxygen to be present for cellular respiration to occur. We'll also talk about fermentation, which is what's going to happen in the absence of oxygen. So it's kind of an alternative form of energy extraction in cells if oxygen is not present. And then we will also, at the end of the chapter, just briefly talk about how some of these metabolic pathways could be regulated, kind of taking this back to our discussion in the last chapter about enzyme regulation. All right, so really what we're talking about here is extracting energy from fuel. And when you see that word fuel, you probably think of something, like gasoline, right? That would be the common fuel that we're using to power our vehicles. In living cells, the preferred fuel is the sugar glucose. Okay, so we can extract energy from glucose. And just to remind you what glucose looks like, you don't have to memorize the structure, but it's a 6-carbon sugar. It could exist in the straight chain form here shown on the left, but in the aqueous environment of your cells, it's generally going to be circularized and look like this image on the right here. So 6-carbon sugar glucose. And we're talking about metabolic pathways here, and so just to remind you, right, metabolism is all the chemical reactions that are happening in the cell, and these metabolic pathways are generally consist of lots of enzyme catalyzed reactions that are all linked together. So we're going to see some of that going on here. And because these energy reactions are so important for cells to be able to function and stay alive, these pathways that we're going to discuss here are really present in all organisms. So every living cell is going to have these reactions or some version of these reactions taking place. As I mentioned in the very first slide, a lot of these reactions are taking place in or around the mitochondria. So we'll refer back to that, but just keep in mind, right, the whole point of having organelles like mitochondria is it provides kind of a specialized structure that allows, these specific pathways to happen more efficiently and more quickly. If we think about this in overview, really the whole point of these reactions is, again, to start with our glucose here, C6H12O6, and try to extract some of the energy from that glucose. And to do that efficiently in our cells, we need oxygen to be present. We'll talk about this more later on, but this is the whole reason we have to breathe oxygen, get it into our cells to allow this to occur. And then as a byproduct of extracting that energy from glucose, we're also going to produce some carbon dioxide, right, which we get rid of. We exhale the CO2, and we'll also just produce some water molecules as well as a byproduct. And what we're doing with that energy that we extract from glucose is generally trapping it in ATP. So, the analogy I like to use is, if you wanted to walk outside the classroom here in the hall and, buy a soda from the soda machine, but all you have is $100 bill in your wallet, you really need to make change first, right? So you can think of ATP as that change, right? It's that energy currency that all these processes in the cell are optimized to use, right? I mean, yeah, you could walk into a store and buy some small item with your $100 bill, but the cashier is probably going to be annoyed at you because they have to give you all their change out of the register and it's going to take longer. Whereas if you walk in with that exact change, it's going to be quick and easy. So think of ATP as that exact change. So we want to convert our glucose, our $100 bill, into that easy to spend ATP currency. Tying this in with our discussions of some of those energy concepts in Chapter 8, you might remember we talked about delta G's, changes in free energy for chemical reactions that are occurring inside cells. And I realize you don't really have a reference point here, but the delta G for cellular respiration for this process of breaking down glucose and extracting the energy that's contained in that large sugar is negative 686 kilocalories per mole. And so the whole point here is that's a large negative number. And you might remember from Chapter 8, if you have a negative delta G, a negative change in free energy, that means that reaction, or in this case, this series of reactions, is releasing energy. So highly exergonic. And again, we're going to use that released energy to drive an endergonic reaction, an energy requiring reaction, which is the production of ATP. Now, there's really three main processes that we're going to talk about in this chapter. So one is glycolysis, which we'll go through in this section, or in the next section, sorry, which is where we start to break down that molecule of glucose and we convert it into a little 3-carbon sugar called pyruvate. And glycolysis is really linked to both of the other two steps on this list. Cellular respiration, which is by definition is an aerobic process, which is where we're really going to extract the rest of the energy that's still left in those little 3-carbon pyruvate sugars. And this is really where we're going to generate the majority of our ATP. If we don't have oxygen present, we'll carry out fermentation instead, which is basically an extension of glycolysis. I'll explain this later on, but it does allow us to make a little bit of ATP in the absence of oxygen. And as we'll see, we also get some other just byproducts, either lactic acid or ethanol and carbon dioxide, depending on the cell type. Another way to look at this, so here's our sugar glucose that we're starting with. Through this process of glycolysis, we're going to break that down into these 2 molecules of pyruvate. We'll take our 6-carbon sugar and split it into 2 3-carbon sugars. And just to remind you, we are getting these glucose molecules ultimately from photosynthesis, right, from plants. So either we eat the plant that made those sugars, or we're eating the animal that ate the plant to get the sugars. But either way, they're ultimately coming from photosynthesis, which we're going to look at in the next chapter. But once we've generated that pyruvate, we're really at this metabolic fork in the road. So if we have oxygen present, we'll go through full aerobic respiration, which will include these steps called pyruvate oxidation, the citric acid cycle, the electron transport chain, and in the presence of oxygen, we can generate about 32 ATP from that one glucose that we started with. Whereas if we don't have oxygen present, if we're in an anaerobic environment, we carry out fermentation instead, and we will only produce a couple of ATP from that glucose. But right now we want to focus on the left side of this equation here and talk about, well, what's going to happen if we do have oxygen, and we'll come back and revisit fermentation later. So as I was just mentioning, if we do have oxygen present after glycolysis, we'll go through these three key steps listed here. So pyruvate oxidation, the citric acid cycle, sometimes also called the Krebs cycle, and electron transport. And as we were just saw on the previous slide, this can generate a large amount of ATP from that glucose that we started with. And as we're going to see, a lot of these steps involve what are called redox reactions, and that's short for oxidation reduction reactions. You probably learned about these in a general chemistry class. And the bottom line is oxidation reduction reactions refer to reactions where molecules are gaining or losing electrons. And usually these will be paired together. So if a molecule loses an electron, which is what we see happening here with this compound A here, it loses an electron and it becomes oxidized. So loss of electron, you become oxidized. And what happens to the electron? Well, typically it's going to be given to some other compound that was already oxidized. In this case, compound B will accept that electron and it becomes reduced. So if you lose an electron, you become oxidized. If you accept an electron, you become reduced. So oxidation reductions paired together. And as you'll see in this chapter, we're talking about losing and gaining electrons, but a lot of times these electrons are being moved around in the form of hydrogen ions. So we'll see hydrogens moving around, but don't let that confuse you. So if we think of this in terms of cellular respiration, what we're going to be doing essentially is we're going to be stripping electrons away from our sugars, so glucose or some of those smaller breakdown products that we generate from glucose are going to become oxidized. So we pull electrons out of our sugar and we're going to give them to other molecules, primarily to molecules called electron carriers, which kind of makes sense, right? They're going to carry these electrons for us. And sometimes you'll also see these referred to as coenzymes, but I'm going to stick with the electron carrier nomenclature here. And the primary electron carrier you're going to see in these biochemical pathways is one called NADH, or NAD plus in its oxidized form. And it has this big kind of scary chemical structure, which you don't have to memorize, but really we're just going to kind of focus in on the business end of the molecule up here, which is where the oxidation and reduction is taking place. And so what you see on the left here, this is the oxidized form of that electron carrier, also referred to as NAD+. And once it accepts electrons, it gets reduced and becomes NADH. And as you'll see from the arrow down here, right, we could also strip electrons away from NADH and convert it back into NAD+. So that's going to be a common occurrence here. You'll see this happening over and over again. NAD+ will accept electrons, get converted to NADH. NADH will give its electrons to something else and get converted back to NAD+. And I also just wanted to point out that there is energy being stored in the acceptance of those electrons. So, not as much energy as would be stored in an ATP molecule, but just the ability of these electron carriers to take on those electrons is a form of stored potential energy. All right, so you might be thinking, well, okay, as you're going to see, there's a lot of detail in this chapter, but what do I really need to know for the exam? And so what I want you to know is big picture. So we'll look at some of the details, but I really want you to know, well, how do these different pieces of cellular respiration fit together? So, for example, this diagram on the left, you're going to want to know. So, we start with glycolysis. The product of glycolysis, pyruvate, leads to pyruvate oxidation. The product of pyruvate oxidation gets fed into this biochemical pathway called the citric acid cycle. Some of the products of the citric acid cycle get fed into the electron transport chain. So I definitely want you to know that overview. What are those pieces? How do they fit together? And I also want you to know, for each one of these pieces, what goes in and what comes out. So for example, for glycolysis, what goes in is glucose, what comes out is 2 molecules of pyruvate, and as we're going to see in the coming section, we're also going to get a couple of ATP, we're going to get a couple of reduced electron carriers out of glycolysis as well. So for each of these sections, what's being produced? Are we making any ATP? Are we reducing any electron carriers? If so, how many? Are we making any molecules of carbon dioxide? That's really what I want you to know here. And we will also come back and take a look at fermentation later on, but for right now I want to focus on the left side here. The other thing I want you to know is where in the cell are these different parts of cellular respiration taking place. And, as I mentioned earlier, this is all happening in or around the mitochondria. So as I go through these parts, I'll try to make clear exactly, where this is happening. If it's in the mitochondria, what part of the mitochondria. So what goes in, what comes out, how do the pieces fit together, and where are they happening? So I'm going to end part 1 there.